Hello everyone. So today we are going to talk about one of the important topic in the microbiology, that is the mycobacteria. So the mycobacteria are the gram-positive acid-fast rods. There are various types of mycobacteria. Those are Mycobacterium tuberculosis, Mycobacterium avium, Mycobacterium saccharophilacum, Mycobacterium marinum, and Mycobacterium leprae. So these are five mycobacterium that are very important, and the Mycobacterium tuberculosis and the Mycobacterium leprae are the two important mycobacteria that we should know in the clinical wise also. So various uh, multiple choice questions and various questions are asked from the Mycobacterium tuberculosis and Mycobacterium leprae. So today we are going to talk about the Mycobacterium tuberculosis. So the Mycobacterium tuberculosis causes the tuberculosis. So it occurs due to the um, inhalation of the Mycobacterium tuberculosis from any infected person. So what are the various symptoms of the tuberculosis? The various symptoms are fever, night sweats, weight loss, cough. The cough could be productive and non-productive cough. There could be the hemoptysis. So now let us talk about the pathophysiology of the tuberculosis, how tuberculosis occurs and how it progresses um, throughout the time. So after the bacteria inhalation, the inhalation of the bacteria can occur from the infected person. There is bacterial dissemination from the infected person, and after we inhale the bacteria, there is the formation of the gone focus. And and after there is formation of the gone focus and there is involvement of the hilar node, we call is as a gone complex. So you have to know here, the gone focus is only the focal lesion that occurs in the lobes of the lungs, but when there is gone focus along with the involvement of the hilar node, then it is known as the gone complex. So this all becomes the primary tuberculosis. So what happens to those primary tuberculosis patients? Those primary tuberculosis patients, more than 90% of the patients who are immunocompetent, the lesion becomes fibrosed and calcified. But at the meantime, if we do the protein purified derivative test, or we also call it the tuberculin test, it is positive. And if gets reactivated, if the primary tuberculosis gets reactivated, then it progresses to the secondary tuberculosis. And if person is um, immunocompetent, then there can occur the fibrocaseous cavitary lesion, and it usually occurs in the upper lobe. It usually becomes the locally destructively disease. It doesn't travels or it doesn't metastasize to other organs. It becomes locally destructive disease, and in those local destructive disease we see the cavitation, we see the cageation, and we see the scar formation. But in the less than 10% of the patient who are immunocompromised states, like the immunocompromised state could be the person can be suffering from the HIV/AIDS, or the person can be suffering from the cancer and is taking the can be taking the immunosuppressant drugs. For those type of patient, from the primary tuberculosis also there can be the bacteremia, and from the secondary tuberculosis also there could be the bacteremia. And after the bacteremia there occurs the miliary TB. The miliary TB means the TB, the bacteria travels widespread through our body, and it infects various organs of our body, and there can occur the TB meningitis. If there is involvement of the vertebra, then there can occur the POTS disease, and there is also involvement of the liver, spleen, and adrenal gland. Now let us talk about laboratory diagnosis of the tuberculosis. We can directly visualize the bacteria by the acid fast stain of the sputum, and also we can do the gene export. We can also do the tuberculin test. We also call it the protein purified derivative test. and the lesion, there is formation of the wheel and there is formation of the erythema in the protein purified derivative test. And if the induration is 0 to 5 mm, then it is not significant, and if the induration is 5 to 15 mm, then there is the suspicion of the tuberculosis, and the induration is more than 15 mm, then it gives the confirmatory result. So what is the character of the secondary tuberculosis? We see the characteristic secondary tuberculosis is that the lesion we see is there is the caseating granuloma with the central necrosis, and we also see as the Langhans cells. So this is the characteristic of the secondary tuberculosis from the lesion. So what is the treatment? We give the anti-tubercular therapy. We give the isoniazid, rifampicin, pyrazinamide, and ethambutol. The anti-tuberculosis therapy may differ from the country to country, and the duration and the dose may differ from the age and the policy of the country. So this much for today about the tuberculosis. I hope you understood the topic on the tuberculosis, Mycobacterium tuberculosis. Thank you.